Hello.
I think sir has joined. Good afternoon, sir.
Good afternoon.
Welcome to Nagpur and welcome to Gurunanak in this STTP online program.
Thank you. Thank you. I mean, I need not have to be welcomed in Nagpur. I'm from Nagpur.
Yeah. Yeah, that's true.
Fine. I'm at home, so I'm at bit casual attire. So please excuse me for that.
Yeah.
And, tell me, how do we proceed?
So no issues, sir. No issues, sir. I think we will start, ma'am.
Okay. So we'll start with the session. Good afternoon, all of you. Myself, Vaishali Kilor. Welcome you all for third session of the fifth day and phase 3 of AICT sponsored short-term training program conducted by Gurunanak College of Pharmacy, Nagpur on the topic to train the trainers for entrepreneurship development in the field of pharmaceutics. I take this pride to introduce you all to our today's eminent speaker, Mr. Prashant Deshpande, who is highly experienced faculty in the field of pharmaceutics. Mr. Prashant Deshpande has an extensive experience working in almost every important division of well-known pharmaceutical industries. Presently, he is head Center of Excellence, Cipla Limited. He is alumni of Department of Pharmacy, Nagpur. We are very proud of you, sir. He started his career in pharma industry since 1987 without any break till today. He has started his career from FDC Limited and worked at various key positions in IPCA Limited, Hext Marion Russell Inc., Cipla, NGC Limited Lagos, etc. He has successfully managed production, plant management and engineering, international regulatory and safety audits, technology transfer and operations, etc. He has participated in number of strategic projects, including Greenfield and Brownfield projects, new platform technologies and skill development initiatives. He has attended coveted GPHA GDUFA conference at Washington and ISP International Seminar on Technology Transfer at Philadelphia, USA. I have his biodata with me and I know you, sir, very well. It is so extensive that time will not permit me to read it here, so I have just shortened it.
Thank you very much
So,
for good work.
beg your pardon. With this brief introduction, I request you, sir, to start your session. I welcome you for the session. Over to you, sir. Thank you.
Thank you. Am I audible?
Yes, you're audible.
So I have only one request. While I'm not a professional teacher or who is very well versed with delivering a lecture like you. I'm very proud of you that you have been teaching the students, bringing up the new lots and the young lot of pharmacists as an input to pharmaceutical industries. I will try my level best how best I can deliver. And second thing, what I want, I am not having a very long presentations, and I'll be happy if I have a number of questions. I don't know how many participants are participating in this. I cannot make it out, but whosoever are there, please be open and don't hesitate to ask questions, because it's always said that there is the gap between an industry and an academics. And I realized that, and I have realized that a number of times. Neither industry is going close to academics, neither academicians are ready to close to industries. And everyone, like an industry, should take two steps forward. At least academicians are expected to take one step forward to at the bridge the gap which we are experiencing since my generations. I've been in industry for like more than 30 years, and I can see that that gap has not at all reduced.
Yeah.
And that is what is my thrust always. I have a feeling that I should do something to bring this as close as possible, and that's the reason I've been participating in this skill development program. I'm associated with BITS Pilani and many open universities, like I have indicated that. Having said so, I just wanted to go quickly to my presentations. Please, if I start talking, if I'm overshooting my time, please raise a hand.
Itadwar sir has also joined, sir.
Oh, very good.
Yeah.
Yeah, bhai. Good afternoon.
Yes, good afternoon, Prashant. And thank you for joining.
Thank you for you also for giving me an opportunity to interact with your faculty and the students.
Actually, it was our pleasure
Yeah.
that you have joined, and your valuable guidance will be useful for everybody.
Are you able to see the present?
Please.
Yeah.
Yeah, I can see it. Please put it on presentation mode.
Yeah.
Next. So let's see why we technology transfer. Basically, this word was not there when we were studying pharmacy. This is the term which has normally been coined and in the industry for last 5 years or maximum 7-8 years, not prior to that. However, its need is always been failed, because we always, when we say that there is a gap between industry and academician, there is also a gap between a development chemist, a pharmacist, and a manufacturing chemist. Both of them, I myself is a man who has worked in a production and operation. I always feel development person does not develop the product. He does not know anything about the product. He does not know anything about the molecule. He does not know about the requirement of a production. And the least can go and go and go and always feel that they are not putting sufficient time, energy, knowledge, and efforts to develop the product. This I'm telling you, since my beginning of my career, even my boss used to say it कि उन लोगों को कुछ समझता नहीं है, बराबर product develop नहीं करता है. And probably we have developed in that culture. However, when I have spent around 20 years of my life, I found that this is the gap we again want to bridge it. So why technology transfer? It's a transfer of knowledge and technology. What is being transferred is often not really technology. What has been developed in a development lab is the state of a knowledge, which in fact is a precursor of a technology, because technology is where it is available only in the plant. It's available only at the manufacturing plant. R&D does not have the set of equipment. They have no uh, what mean or equipment, set of equipment for a process development. So their focus is always on a product development. They are never putting their efforts for a process development. And when we say in a pharmaceutical, both product and a process development are critical in developing a product. Technology transfer as of today is an indicator of manufacturing excellence. When we develop a product, when we file the dossier, and when we are able to launch the product in a seamless process, that means that product is developed as based on a QBD approach. It has uh, passed all stage gate of product development, and it has successfully, the technology and knowledge is successfully transferred. This happen very rarely in the industry as of now. So there is a lot of things which is expected from an development team. There are many CMOs like contract manufacturing, and there are multiple number of CROs which are taking this as an opportunity. So somewhere when we are saying an entrepreneurship, this is one of the opportunity wherein you can explore to have your contract research organizations. When we say contract research organizations, there is an opportunity to develop analytical method transfer, which goes along with the product development. When we say that we have a CRO, we have a different excipient, set of excipient, which is ready to use sort of a formulations. Like, Opatra is the film coating. There are some set of equipment excipients which are, processed excipients. When we mix, we can develop the DC formulations, and so on and so on. So there are a lot of opportunities in the field of CRO and related development activities. Next. So I'm not going to read this, but I will give the I will keep this slide for you to read. The US FDA guideline also emphasizes the importance of technology transfer, and the emphasis is on a practice of transferring the knowledge and the scientific findings, whereas the WHO guideline further elaborate it to have a logical procedure and the controls which is required for the technology transfer. Same is true with ISP. ISP also mentions about a systematic procedure for a technology transfer. If in a technology transfer is like a relay, basically it's an baton. It's a baton from one hand to another hand. And when we have a transfer, there has to be two entity. And that's the reason subsequently when I will say the organogram and the other things of a technology transfer, I always emphasize that technology transfer should have an independent identity in the organization. If they are with the development team, they are biased with the development team. If they are with the production team, they are biased with the production team. And very nicely, Geoffrey has mentioned, research transforms money to the knowledge, and technology transforms knowledge into the money. So it is a link between the research and the commercial aspects. Next. So this is next. This is a typical legacy development to commercial process. The R&D blocks develop the product in grams and kgs. Analytical method is developed based on the batch or a couple of batches which is developed. Finished product specification is defined. Method validation is still in the progress, and a product get transferred to the plant. Okay, plant execute feasibility batch, one assessment or optimization batch, and a regulatory batch. So in all, the plant has a maximum experience of 2 or 3 batches. Typically the batch size is 50 kg. We are talking here the registration batches, and the same batches has been tested, released, and the dossier get filed. The specification is based on the batches which is executed at a development, which is maximum of 1 kg or 5 kg. So the in-process parameter, the process parameters, the critical quality attributes, the critical process parameters are all defined based on that 5 kg batch size. And when we execute the feasibility optimization registration batches, many times, since that is closely monitored, the parameters are more or less get complied, and because it has a broader parameter window, and the dossier get filed. However, when we go for a launch for a 500 kg, we found that the parameters or critical quality attributes or dissolution parameter or dissolution profile are not complying what is expected out of development or out of registration batches. And then we start digging out in an investigation forum and a kappa and deviation and blah 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 blah. It becomes, the meetings after meeting. How do we communicate? How do we justify? How do we change? What justification? What rationale we can give? And we lost the market, because the regulatory market is such that if you develop the product, if you launch the product in time, you have all the revenue. If you are losing that opportunity, you lost the market, you lost the margin, and you lost the business. Nobody is waiting for you. There are plenty of players there around to play with the same molecules. Next, please. If anybody has a question in between, please, it's open. Don't be shy on asking questions. So what are the issues and opportunities? Maybe this presentation is made or extracted 4 or 5 years ago, when the things were really wanting. In fact, we had a lot of challenges that time. When I say we, I'm saying overall all industries in India. There was a limited involvement of the operations people and the other people in the development journey, because it was very closed window activities. Nobody was allowed to enter into R&D. Many of you must be knowing that, to have an entry into an R&D lab or laboratory also required the permission from the management. Why you are going, where you are going, because of intellectual property protection or whatever reason. But it was very restricted moment area. And that's the reason probably the involvement in the development journey was very limited. Analytical method development is a very important and a critical aspect. I always emphasize throughout my career, in my practice as well as in discussions, which never go hand in hand with the product development. It is always lagging behind. And the method they develop is many times is not discriminatory. What it means, discriminatory method, if you have a particle size method of analysis, which is by Malvan, or which is by other instrumental method, laser diffraction method, it should always be discriminatory method of analysis. That if there is a variation in the lot other than what has been prescribed in the limit, it should be able to identify. Similarly, a dissolution method, if you change the source of in excipient, critical excipients, or if you change the source of active ingredient, if the particle size of the API is changed, or if the polymorphic form of API is different, then your analytical method should be able to identify it. And it should be able to identify that, the results which we are getting probably is because of this factor, and which is normally not happening. So I am saying that I'm not saying they are not doing it, but their pace, the progress is not going hand in hand with the product development. And then nowadays we have a lot many effective stage gate process. In fact, there are 9 stage gate process of a product development before it get registered and launched. And every stage gate has to be signed by a respective cross functional team before we go ahead with the product development and our registrations. Normally, I have a complaint that the product development does not consider manufacturability. The factors which are important in the manufacturing. So granulation endpoint, when we say typically OSD products, when we do a granulations, the rate at which the binder has to be added, the time for which the binder has to be added, the time for which we give a sufficient time between the binder addition and the subsequent water addition, the time factored in, considered by a development team, which is a part of a process developments, are not put it on it's never there on the checklist for them. It's for them, it's very simple. Develop a product, test it for assay, test it for dissolution, test it for related substance, and for them the product is ready, which is normally not we are not able to photocopy it in our manufacturing plant. There are few processes which cannot be validated. There are few processes which cannot be automated. We are talking about PAT tools. We are talking about seamless, paperless process. But as of today, I'm talking in the year 2020. Pharma industry is not even 3 sigma, and what we want is, the automobile industry is the aeroplane industry is 6 sigma, 1 in 1 million. That's the failure rate there. Whereas we, as of today, we throw crores of rupees because the product is not complying to the specifications, and we don't know why the reason. We are not able to pinpoint the reason why the product is failed. We are not able to define a kappa for that failure, and that's the reason this is very important aspect. I will probably repeat this sentence. Next. And probably why this is so, that we are also not giving sufficient resources for the product development. We are not giving sufficient time for a product development. The market is very competitive. The timeline is always against us. Everything in industry, they always want it yesterday. Even today is a late for them. So they have a very limited resources. They have a very limited timeline. The learning and experience, the QBD is too big for them to comply, and development to production, the knowledge sharing is also limited. There is always saying that, just share what is required for the production. Don't share total development path with the operations people, because they will evaluate, they will interpret all the facts in their own way, and that will create more problems. So keeping the knowledge in a closed door or window, or having a limited access, always create a more of a problem, because the failure which is normally experienced in the development, we get it after 10th or 11th batch. And then we realize that this was an observation, even a development we have neglected, we have not shared. However, now it's difficult for us to address it and close it. Then, between development and a plant, we cannot do any development activity in the plant, and the development people are busy in developing a new product. So when the legacy or existing registered product has a problem, when we want to improve the process, when we want to improve the product profile, when we want to do any AVD, we don't have the facility. So that's the reason there has to be a one pilot plant, wherein this test hypothesis, why the dissolution has failed. Is it because of an overgranulation? Is it because of an overdrying? Is it because of a higher hardness? Is it because of some other reasons? Those hypotheses can be tested. Next. I think I've covered most of it. In India, as of today, though, statistical tool, we have started using it, like an at least organization like us. I will not take a name, and the allied industries, rest of the N-2, N-3 category of industries, they are not utilizing statistics, because when you develop a product, whatever 5-6 batches you develop in R&D, if you put it in all the available data systematically in the statistics, and do statistical evaluation, and then define the process parameter window, in process parameter window, or critical quality attributes and the limits for them, many times probably we will be able to fix the problem which may get encountered in future. So statistics is an important field which all pharmaceutical education and academician should teach to the student. Statistics is not only mean media mode. Statistics is not only a probability. Statistics is much more advanced statistical tools are available, and probably we may not be knowing it, but those are now available, easily available, accessible. Those should be taught to the students as in one of the topics. Very, very critical across all pharmaceutical streams. Let it be clinical pharmacy, let it be analytical, let it be production, let it be other set of our subjects and the fields also. Next. Yeah, this is what we are missing. I'm just trying to say this is how we are production and the development team. Both of them work hard, but two end never meets because they are not aligned. Next. I think the same thing. Transfer of a technology requires a planned approach using a trained, knowledgeable person working within a quality systems, with a documentation of data covering all aspects of development, production, and quality control. Next, it's a teamwork. This is typical organogram of technology transfers. So here in my model, I'm saying that he should report it to plant. He should not, and some of the industries like our competitor industries, the technology transfer lead reports to development team, and I'm not for it, because the one who is a giver cannot be taker. He should always be linked between a plant and development. And I'm broadly put the activities they are in new product transfers, they are in new launches, they are in they are actively involved in validation, execution of validation batches. They do a gap analysis after, when we execute the batch registration batches, we find the dossier, and when we get an approval, this is a time which varies from say a year or a 2 year, and during this 2 year many things get changed. So when we get a product approval and when we are required to launch the product, we found that there are a lot of gaps. So this is the time we do a gap analysis, and we identify the gaps and we close the gaps when we go for a launch. And the tech transfer has a lot of things to play around during this activity. Site transfer, continuous improvement, ABD, investigation and troubleshooting, and the relevant documentations are other aspects wherein the technology transfer person are playing their roles. Next. So there are two parts. How it was a couple of years ago. I'm talking about my organization, how it should be. So initially the R&D used to develop the product, they used to do a pilot, they used to execute the registration batches. Nobody from the plant used to monitor it, and suddenly scale up validation. When the development people have detached, it becomes a baby of a production and manufacturing thing, and there was no linkage, there was no overlapping. What is required isn't there is an overlap, that the product is being developed and the registration batches are executed, and technology transfer is participating in activity from a pilot stage, and it's taking over till commercial, and manufacturing is a part of technology transfer. I've put it in the bottom footnotes. What is R&D's deliverable? What is technology transfer team's deliverable? And what is manufacturing team's deliverable? So the knowledge transfer is smooth and seamless. It's not detached. It's continuous. Next. Am I going a little fast? Is it okay?
Yes, sir. It's okay.
Are you comfortable with the way I'm going?
Yeah.
So again, the 9-stage get process is put into the blocks: IPD pilot, then a technology transfer team, and a production block. Then, these are the stage gates. Strategy review meet, pre-formulation studies, formulation review meet, analytical method development for process, tentative specs, which is getting defined here in IPD, initiation of QBD studies to determine design, space, and control strategy. In the other form, we start risk evaluations. What are all our risks? It's earlier we used to do risk assessment at the end of a process. Now we have started doing it during the development. The second block is critical quality attributes, critical process parameters, then the confirmation and the pilot. Now, earlier we used to carry out direct bioequivalence study. Now we have a pre-BE study. We do it on the smaller set of subjects, depending upon the class of the molecule, it's as per which BCS class, how the innovator sample is behaving in the dissolutions. Based on the literature survey, a small B study has been carried out on the IPD batches. And then the larger scale, full scale biostudy, biostudy has been carried out. Now, even the protocol is shared with the regulatory authority, and the consent of that is taken. Because if we earlier we never used to take such consent, and then we used to have the queries, and responding that query was always a difficult. So proactively nowadays, the organizations are interacting it with the regulatory bodies and sharing it, the strategy, how the product is developed, how the product is going to be analyzed, what is an analytical method, what challenges we have, how we are addressing it, how we are going ahead with the biostudy, what subject, what protocol, and what risk we are carrying, and how do our what's the risk mitigation plan we are designing. So when the dossier is reviewed, he is knowing that these are the bullet points which he needs to review. He checks it, he ensure that they are properly addressed, and that's the reason we started getting our dossier approval. And some of the products are even getting approved less than 1 year. Earlier the approval timeline used to vary from anywhere between 2 years, 3 years, 4 years, 5 years. Some of the products has been approved after 5 years, and 5 years is a too big span for the between the product development and commercialization. Can we go next? It's mostly I have kept it on OSD, but I also have on other doses form because the requirements are different for a parental doses form, for topical doses form, for liquid oral doses forms. Probably same thing has been explained here as a flow. I have already shared the PPT.
Hello, sir.
Yeah,
Sir, I have a question.
yeah.
So, as you said, like in pharmaceutical industries, like the Three Sigma limits have been used most probably, and in the aeronautical and aerospace, they use Six Sigma limits.
No, I'm not saying that. Yeah, what I mean to say here that the failure rate
Okay.
in automobile industry is 6 sigma, 1 in 1 million.
Okay.
Whereas we are still at 2 or 3 sigma.
Three Sigma.
That means the rejection rate is high.
So, why we are not going up, like to reduce the rejection rate, like by using bell curve or normal distribution method?
It's not statistical evaluation. It's an target. We are at 3 sigma and we want to go at 6 sigma.
Okay,
That
but
means my one batch in 1 million I can accept as a failure.
sometimes a QBD in Six Sigma have a diminishing returns because they have been not even reaching the Six Sigma, and will continuous having DMAIC based projects. So that will be a problem sometime for pharmaceutical manufacturing products.
That is what is our challenge is. That is what is we want to close it. That
So
is what
how,
that is.
okay, so how can we try for that? Like as you said, so how can we try further, like applying quality by testing method, producing, checking with the variations and all?
By putting more methodical efforts, by using all available tools, by
So it's,
improving our approach, development approach, by successful technology transfer, by improving manufacturing capability,
okay,
by
so
improving
it's,
the technical capability of a person.
so it's like re-engineering, trying again and making it possible to happen.
Not re-engineering, it's basically a continuous improvement. We
Okay,
are improving and improving, and in improving we are far away from our goal.
okay,
Think over it. If I am throwing crores of rupees, I cannot give you a figure. Hundreds of crores of rupees with no reason, and it's not across industry, across globe. You know how many recalls go to the US FDA website. How many recalls are there? It's a sheer waste. If those recalls can be stopped, if you are able to identify the root cause of that failure, if you are close that, if you stop those recalls, the cost of product will reduce, the margin will increase, and access to the drugs will be better. The product cost will reduce. We probably we may get paracetamol at 50 paisa. If it is 50 paisa, we may get it at 25 paisa. I'm just giving you an example. If we able to close and reduce that rejection, a percentage of rejections, I don't know whether I could explain you. Are you satisfied with my answer? Hello.
Sir, sir, can I ask one question?
Yes, please.
Sir, there are certain instances where we have to develop the analytical method. Say, for example, 4 or 5 years back, I have worked with teicoplanin and serosopeptidase. So, in that case, the analytical methods were not there in IP, Indian Pharmacopoeia, and we had lot of problems because no analytical method was there, and we were trying to develop the method, but then whether it is correct or not, to whom we should contact, to whom we should contact for standardization of the method, that we do not know. And then after the work was over in 2017, then it published in Indian Pharmacopoeia. So in such cases, what we should do?
Normally, for analytical changes in analytical method, of course, we go analytical when you develop an analytical method, you have to do a method validation. And we all know there are 6 chapters of method validation. Robustness is there, and linearity is there, and there are other chapters are there. Once you validate your analytical method, you can very well approach pharmacopoeia that this is the method. It is complying all the chapters of method validations, and this is what is we want to publish it as an official standard method. Even if the pharmacopoeia has a method which is published in a pharmacopoeia, if you have a method which is alternate to that method which has been validated, you can still
Okay.
approach pharmacopoeia and tell him that we can request that put this as an alternate method. So
Okay.
that's the reason you have a method 1, method 2, method 3, method 4. Sometimes you will find that those 5, 6 methods has a different limits. So based on the method, the limits are also different, but they are accepting it. So Indian Pharmacopoeia Forum is there wherein you can approach and they do reciprocate. They do revert back on if you communicate with them. Similarly, US Forum is also there. Similarly, BP Forum also is there. So if you write
Okay.
method transfer. Normally in India, analytical team looks for the approaches. Which base method kahin hai kya? And many times we found that the so-called as patented product or a registered product or a NCE molecule, their analytical method is developed only for their product. They keep things so tight that if you put your product into their method, we encountered many challenges.
Yeah.
The forum gives even the US FDA. I'm not able to recollect the name. There is a site wherein we get the analytical method, reagents, and dissolution, but the information is very limited for us to work on it. They don't give you a complete, and that is their property, intellectual property.
Yeah.
That's their business.
Yes.
We can go next. If you are, yeah, you are ready. The responsibility in a job description of a tech transfers. It supports R&D for the introduction of a new product. It supports in product launch. Normally, when we say product launch, when we get an approved, we get an order of 50 batches. We get an order of 100 batches. The launch is always big, very big. And those number of batches, 25, 50, depending upon the product, are required to be kept ready, that we are expecting a product approval and this product will be launched. If any of the product get OOS out of specification deviation, our launch is required to be deferred because we need to have the reason and a kappa defined to it. If you are not able to identify the root cause, if you are not able to define a kappa, if you are not able to file the variation, if you are not able to get a variation approval, our launch is deferred. All 25-50 batches may go west. So it is very important that we have the product developed as per QBD, the technology is transferred as a seamless process, and we file the dossier and we get an approval and the product executed as per the dossier, we are able to launch it and commercialize it. Technology transfer team participates in process development. Process development is a continuous process. We change the mill. When I was working, we used to work on a multi-mill. When I was working, we used to work on a CAD mill. When we used to, and the multi-mill has an RPM of 300, 400, 600 RPM. Nowadays, we have a co-mill, quadro mill, which runs at 3000 RPM, 4000 RPM, 5000 RPM. The throughput has 10 times more. The equipments are different. The RMG design is changed. I was granulating ultragen analgen tablet. That time we were having what we call as that blender mixer, horizontal blender mixes. Then we have a PLM. Then we have an RMG. Now we have a fluid bed dryer. We have a fluid bed processes. We have a different sets of roll compactor. We have an hot melt extrusion. We have a hot melt granulations. So equipment have changed. And it's always better to move from this old set of equipment to new set of equipment because the throughput is increased, the footprint is reduced, the product is more stable. And when we used to granulate it in a PLM and dry it with 1:1 ratio of water, the impurities were more. When we have moved it to RMG, the impurities has reduced. When we have to move, when we have moved from RMG, rapid mixer granulator, to fluid bed dryer processor, our dissolution profile has improved. When we have moved from this set of equipment to roll compactor, our product stability has improved. When we are moving from this equipment to hot melt granulations, our footprint, which was taking a big granulation or drying suit of say 10 meter by 10 meter, has reduced to now 2 meter by 2 meter. That's giving the same throughput what my 1, 2, 3, 4, 5, 6 set of equipment was giving. So the things are improving, and this process of improvement will always happen in any industries, and so is happening in pharmaceutical industries. We have to harmonize our formulations. Many times it happens that the product developed from because we merge and demerge. We buy the dossiers from the other industries. We take over and the company is buying some other organizations, and the product are also moving along with those mergers. So we harmonize their formulations with our formulation, and the selection of a correct, easier formulation is the continuous process in the industries. Alternate vendor. If somebody has a macrocrystalline cellulose which is cheaper and a better and having the better powder characteristics, will go for it because that improves the commercials of the product. Site transfer is always the continuous process. The earlier the plants were there in the Mumbai, the cost of manufacturing was very high. The salary is very high. The electricity rate in Maharashtra is very high. We moved the product to Baddi in North Himalaya. We have moved the product to Sikkim. We are moving product to Goa. We are moving product to Daman. So site transfer is a continuous process. The moment we found that Daman plant is outdated, it's no more complying the current GMP requirement, that is removed from the network, and we call it as network optimization. That is, those plants are closed down, then the products are transferred to new, other, better, productive, improved plants. So this is a continuous process wherein the tech transfer is has a very important role, because he is carrying the knowledge from an R&D to a plant, and one plant to another plant, and a second plant to maybe 1 to 3 plants. Sometimes same product is manufactured at 3 different locations. Sometimes a product are manufactured in all the locations. Like Cipla has 40 locations, and few of the molecules are manufactured in every plant, because logistically it is always helping the organization, that if the product is manufactured in the Sikkim, it is catering the east zone. If the product is manufactured in Baddi, it's catering north zone. So many aspects plays important role in it. Next. Yeah, I think I've mentioned about it. The participation at a major stage gates. It's a 9-stage gate process of product development, which is now widely utilized in pharmaceutical industry. And which starts from a procurement of API, finalization of a batch size, because if we are, and batch size is very important because it is based on the projection by the commercial team. The commercial team gives you the, what you say, portfolio team gives you the projection of the volume and the business. Based on that, the batch size is defined at the time of registration. The reason being that your registration and a commercial batch can have the maximum ratio of 1:10. If my registration batch size is 1 lakh, I can have my commercial batch size of maximum batch size of 10 lakh. But if the market is very big, which is projected by the commercial team at the time of the product development, before even the product development by the portfolio team, I will keep my registration batch as 5 lakh, so that I can increase my commercial batch size up to 50 lakhs, that is 5 million. Yeah. You are on which slide? Yes.
Should I move to previous one?
New product launch. You are slide number. What's the slide number?
I cannot see the slide number.
1 minute. Yeah, fine. Yes. So the requirement which is not there, and now it's been demanded by the plant people, that we need a lab. We need a dedicated manpower. SME here is subject matter expert. This is another term which is coined in industry. The one who knows everything about OSD. He is an OSD SME. One who knows everything about a topical product. He is a subject matter expert of topical product. Same is true for sterile product. Same is true for other dosage form. Earlier, there were only 1, 2, 3, 4 dosage forms. Now there are plenty of dosage forms. Many. Yeah, next. Yeah, this is just I put it here. Next. This is again after we registered what is an optimum batch size, because if we finalize a batch size for commercial scale, we are required to do a validation of a 3 batch, validation of that batch size. We cannot keep on changing batch size every day. That the requirement is more now and the requirement is less now. And based on the commercial requirement and the projection for 5 years, the batch size is finalized for a launch. This process normally starts immediately after dossier filing. The gap analysis is a continuous process which is happening during a filing and approval timeline. In between, we execute an assessment batch. After filing a dossier and a launch, we execute an assessment batch so that we are sure that we are able to launch the product. Next. I think I've talked about this. The tech transfer team is the team who is participating and working along with the lifecycle management. Team LCM team, we call it, which has a multiple role to play, like process improvement. If you have a failure, OOS, how do we address it, identifying the root cause for it, identifying a kappa for it, the CIP process, how do we reduce granulation time, how do we reduce drying time, how do we reduce the blending time, how do we improve the compression and compressibility, how do we reduce compression cycle, how best we can reduce the coating cycle, how best we can improve the yield, how best we can reduce the losses. So many things can happen. The AVD for active, AVD for excipients, moving from RMG process or PLM process to RMG, moving from RMG to FBE, moving from FBE to roll compactor, moving from roll compactor to HME. So this is the process which is happening all across the organizations for multiple products. EVD, I think I have next. Yeah. What are the benefits? We catch the problem early and solve them in a lesser time and a cost. We don't lose market because we are fast to act, we are predictive, and we are efficient. We have a less number of failures. We are moving towards Six Sigma. We are not required to tweak on the shop floor. The shop floor person is confident to operate. His confidence is very important. It is improved manufacturability. He is relaxed. I think this is coming again. I'm sorry for this. Next. Next. Yeah, this is the last slide which I have put is the technology transfer as a site transfers. What are the activities? It's a part of life cycle management. We transfer documents, we transfer analytical method, we do a gap analysis, equipment equivalency between 2 site and 2 plants. We do risk assessment where all we can expect a failure. We have a mitigation plan. We do have a change control procedure. We execute an assessment batch at a receiving unit. We do variation batches. We generate stability data, compile the data, file a variation. Depending upon the changes, the type of variation is defined. Depending upon the type of variation, approval timeline is defined. Post-approval, the process validation batches are executed and commercialized. I think thank you. I took 10 minutes more, but I'm ready to take a questions. I'm not able to read the questions because I was looking at my screen, but I can see a few people have put the questions. So you can read and I will try to answer them.
Any questions from the participants? I cannot see the questions in the chat box also. If anybody has a question, you can put it in the chat box or unmute yourself and you can ask the questions too, sir.
Okay.
Sir, can I ask you one question?
Why not? I mean, that's what I'm saying. Anybody can ask questions.
Yeah. And, sir, if we have developed a product and if we are having the patent and if we want to commercialize it, then what is the procedure or what we should do?
If it is a new molecule,
No, existing one, but with improved bioavailability, say for example, or many advantages, and if it is patented, then how we should go for commercialization?
either we can ourselves do a commercialization, for which you should have the license facility, get the product license, get the plant approved by, if it is for India market, from local FDA, and launch the product. If not, approach an industry, approach the what you say competitor or whosoever is having that brand,
Yeah.
convince them that you have a product which is having a bioavailability better. If it is a niche product, I'm sure there are people to buy. If it is me too product, in India hardly anybody thinks about bioavailability.
Okay.
I mean, to be honest with you. But
Yeah.
if it is a niche product, product for a critical illness, certainly everybody wants the improved bioavailability. If it's metformin, I don't know whether you are aware, 55% of the drug only is absorbed.
Yeah.
So if somebody is giving us improved bioavailability, certainly the people are there.
खिड़की बंद कर.
No issue.
Yeah.
खिड़की
Same
बंद
is true for respiratory product.
Yeah.
काच बंद कर काच.
If you know, we have an MDI, metered doses form. We have an aerosol product. When we inhale the product, not even 15%, 20% drug is absorbed. 80% of the drug goes west. If we improve that from say 15, 20, 30%, depending upon the molecule, even by 5%, 10%,
Yeah.
you are helping patients in terms of compliances of the therapy. You are helping industry by reducing the number of doses, reducing the cost of product, overall improving the economy and the what you can say the margins of the product.
Yeah.
Your product can be sold in a better way, and that is where all MDI and DPI, what we call it as dry product inhalations or emitter dose inhalations, how do we improve the bioavailability of the respiratory product is the research focus.
Yeah.
There is no model available. The lung only BARC has the lung model available.
Yeah.
Alveoli and alveoli and alveoli and alveoli. When the drug goes to the last compartment of alveoli, microscopic. It's hello.
Sir, there was
Yeah,
some connectivity issue, I think.
and the challenge is that even we do not have the robust analytical method of analysis for particle size.
Yeah.
It's not that the method is not defined. Of course, the method also has a lacuna and however, a sampling also is a challenge. If I jet mill my API, and if I take out a sample from the top layer, I get a particle size, and if I take the bottom portion, I get a different particle size. How do I know what's and D90, D50, D10 of this lot of API? And it only reflects when the batch is manufactured and tested in the quality control lab. So analytical method is very, very important because through a diffraction, if you have a long needle, it gives two different sets of reading.
Mm. Yeah.
One is length, another is diameter.
Mm.
If it is polymorphic, different crystalline form, it gives a different reading. So your method also needs to be very robust.
Yeah.
It's always defined for the window. So lots of challenges are there in a simple formulations like MD and DPI. This is never taught in pharmacy.
Yeah.
And analytical method, assay, related substance, dissolution, pH is all been taught, but nobody teaches you about particle size distributions. If somebody is teaching it in pharmaceutics as a micrometry, it's what we say is only a granules parameter. You can say 0.5% of the industry's requirement. There is nothing big in teaching what is bulk density and tap density. You fill the flask, and in a volume versus mass is a density is known even to the 10th class student. But industry's requirement is much more than this. We don't know when we say this man's height is 5 meter, we know 5 meter is this. But if somebody tells me that this API has a lot of D90, say 5 micron, I don't know whether it is really 5 micron or it is 8 micron, because he is saying his particle size analysis report D90 5 micron based on the sample which he has tested. Whether his sample is biased or not, he will say, I don't know.
So where if the particle size is different, say for example molecular size range and colloidal size range, so the method will be different.
Yes, has to be.
Okay.
Has to be. And when we say AVD, alternate vendor, it is
Okay.
always proposed that you use a vendor's method. But if you have a method which is already registered, if you test the alternate vendor samples with your registered method, we found our results are not matching. And the vendor will say, you test it by my method, the result will match. We cannot go with that method, because if I use this method, my earlier method will not comply with the
Yeah.
other vendor. So
So
harmonizing this method, giving the correct result, because that is defining my dissolution profiling.
yeah.
Yeah, I saw a few questions, but I am not able to read them because I'm on the mobile.
I think Keshav sir wants to ask some question. He has raised the hand. Keshav sir, do
Sir,
you?
kappa guidelines are applicable for or during scale up also, from lab scale to large scale,
Yes,
because that's where many unit processes show the changes.
yes. Any failure, you should be able to identify the root cause and you should have a corrective and preventive action.
Okay.
That is what is QBD says.
Yes.
And that is what is not practiced.
Anybody else wants to ask question?
Technology transfer is the topic for which an academic in the academic you, or at least in post-graduation level, we can have a full scale subject of 100 marks.
Yeah.
It can cover the life cycle management, technology transfer, process optimization, scale up techniques, process validations. The validation which is covered in quality assurance is different than what is covered in process. We have a one term which we normally use in industries, particularly DC formulations, wherein our BU fails. Blend uniformity fail. But when I compress the batch, my CU passes. So we call it as BU fails, CU pass. Tell me, how do I justify this? Is this my batch not complying? It's complying because the CU of stratified sampling is perfectly complying with the AV value less than 5 or 6. The limit is 15. But my BU RSD is more than 5, which is the limit. It's going up to 14, 15. The reason being, it's not been sampled properly, and the samples are not been analyzed properly. So how do we sample? This is that, difficult to mix. If somebody is from QA, probably they may be knowing the theory that in the blender they have to identify the red zones, which is difficult to mix. There is no place in the blender which is difficult to mix. Blender is in simplest, less understood equipment, neglected equipment, neglected process in pharmaceutical industry. We take out a sample, nobody knows whether he taking a sample of 1x, 2x, 3x. Half of the sample by transferring to the glass bottle is falling on the floor. And he is transferring those vials to the QC, and QC is analyzing, reporting that out of 10 sample, one has a 50% content. How is it possible? I have put a salt in the chutney. It has to be distributed evenly. How can you have something somewhere 50% of the salt? It's not possible. It's against the theory of mixing. But he says that my result says so. So I call it as you have a syndrome of BU fails, you pass. Now you tell me why it's not been sampled properly and why the samples are not handled properly. Sometimes even they prepare the solution of a blend in the night, and in the morning somebody puts it on HPLC. Who has studied the solution stability of that sample? Sometimes they have a 40 set stratified samples, 40 samples, 50 samples, because every 10-15 intervals they have to take out a sample and test it. So when we do puja, we have been told that put gandha sage kade, phula sage kade, and then somewhere we miss it. So he also miss it somewhere, and that result is 50%. Then he has a syndrome of you fails, you pass.
So sometimes the polymers also swell, and they may also affect the granulation technique and everything. So in
Yes,
that,
but it will be uniform. It cannot be isolated. It cannot be isolated. It is possible if your product is not properly developed. Like some people have I don't know why they develop such product. They put the binder in the dry mix and they do a granulation with the water. Then you can have this challenge. Binder is 5 milligram and you are expecting it to get it uniformly mixed and granulated is not possible. Binder has to be added as a binder.
Okay. So anybody is
Okay.
having any question?
Any
One more?
questions? New questions?
Oh, you are driving?
No.
No problem. You can go ahead and ask.
I think there are no questions now.
Fine.
Thanks for your elaborative talk, sir. It was really very much informative.
I don't know.
Yes, it has
Somewhere
given
the
very
industry close to academic, and academic should go to industry is what is my theme of
yes.
Hand
So
over
we
to
also
Kilon.
wish to reduce the gap between industry and academia, so that
take one step
we
forward.
academicians can get a good insight into the things which you have focused on. That is the technology transfer. It should be a subject for us, because we are having a very little knowledge about it. But by listening to you, we got to know about the importance of it. As well, you told us about analytical method development, how it has to go in hand with the product development of the product. It is always lagging behind. That is the problem. We have to shift from 3 sigma to 6 sigma in pharmaceutical industry to reduce the rejection of the batches. That is also very important. And the very important point was our entrepreneur inclined people can go for contract research organizations where development of analytical method can be carried out for the product which is being developed. So that was a very important point we noted down, sir.
We can have a public testing laboratories, which are very limited in number.
Yes. So I think the participants have taken note of it. We should reduce a gap. We should give a due importance to reduce, to carry out gap analysis before product launch. That was a very good teaching from you, sir. So it was a very great to listen to you, and very interesting. We are very thankful to you, and in future also we would like to hear you. Thank you very much, sir.
All the best. Thank you, Abhay.
Thank you, sir.
Thank you, Prashant.
Yeah.
Earlier, I used to simply evaluate the papers wherein the questions used to be transfer of technology from lab to pilot to industrial scale. And now the first-hand information from you. Thank you. Thank you so much.
Yeah. Thank you. Thank you, Abhay.
Thank you.
So thank you.